big old letters. Tentative lecture. I do tend to give you time. If I get ahead, no problem. And I won't, if you look at the exam schedule, I can only test you up to chapter 5. Even if I'm lecturing on 6 or 7, yeah. But I will only test you up to chapter 5. And I will send out an email as to what the last slide you're responsible for that exam, so everybody should be able to study properly. So remember, chapter 1 and 2, you read on your own, it's still testable, and then what I'm covering. If I get deep time, same thing applies. I only can test you on what I talked to you guys about the previous Wednesday, and same thing for the Thursday class. Whoever I finish the least amount with, that's what's going to be on the exam. If Tuesday gets ahead of you, then you still go by what I finished with you. If you get ahead of Tuesday class, it's what I finished with on Thursday. Okay, so now we got this weird thing that's zipping up. So we had the neural tube zip up. Actually, everything is zipping up at the same time. As the neural tube is folding and sealing and forming this long tube, so is the rest of the ectoderm. It's going to form the skin over the nervous system. And it's going to zip up towards your head, and it's going to zip up towards your rear. You got a tail early on in the first month. You had a little tadpole tail. Eventually that's going to disappear. You see the limb buds, the leg buds, and the arm buds forming. This is really, really early. This is the first month. Mommy may not even know she's pregnant at this time. So it's before she's missed her first period. And if she doesn't cycle regularly, maybe 2 months before she catches on, then she's probably pregnant. Okay? So when you're looking strange like this, you got all these somites developing along the back, and they're going to start sending muscles and connective tissue out. It's going to make axial skeleton, and then it's going to also go out the buds. Up here, you're looking kind of like a fish. You've got gills, you've got pharyngeal arches and pouches up here. This is going to be important when we get into our endocrine system. Things that were up here, are they going to migrate throughout your body just like that neural crest migrated? And these are going to form things like your thyroid and your parathyroid, and your thymus gland. They're going to form different endocrine glands. Up here, we've got forebrain prominence. So, front of the brain, we've got a lens plateau. So, up here in the face region, the front of your eye develops up in this region. The back of your eye has to grow out of the nervous system and come forward to meet up with that lens plateau. If the two don't meet, you don't get an eyeball that functions on that side. You've kind of got this murky, kind of grayish mass there, and no functioning system. So, caudal neural core, cranial neural core. Who has never heard of spina bifida? Okay. This is a birth defect. When you have the simplest form of spina bifida, you get everything neurally formed wet. But you don't get the complete formation of the vertebral arch around the caudal end of the spinal cord. You got a little some freaky reason, you have a little hairy patch at the base of your spine, right over that opening. And that's the only thing that the doctor will see. This freaky little hairy patch, and he'll say, spina bifida occulta. And mommy says, and she goes, no. Unless you get a blow to that area, no problem. How many times are you gonna, well, if you're gonna be a boxer, you're gonna play rugby, or you're gonna play something that's a really heavy contact sport, maybe you wanna get some bone and close that little gap there for protection, but it's not gonna compromise your nervous system at all. But the most severe spina bifida is called spina bifida myeloschisis. You'll see this later on. That term, myelo, is referring to the nervous system, and schisis means to cut. And it looks like somebody came with a scalpel and sliced the neural tube and folded it back open. They didn't fold it back open, it never folded. It never folded, you got major neural tube problems and probably peripheral nerve problems. You don't have to really look for the damage. You're going to see a huge cyst on the low back. It's going to look like a huge blister. You're going to have you're going to have all the meninges out there in this cyst. You're going to have nerves getting pulled out there, maybe spinal cord getting pulled out there. And you're going to see that the feet are all kind of, they don't look like they're supposed to. They're supposed to, babies' feet, they're up here like that. But this, they're not even in that weird position. They're all kind of misshapen. You know that they're not working. You stimulate the nerves, they're not working either. That's the most severe form. And in between, you've got less severe. If you just have a meningeal seal, you have the cyst, this meninges out there, but the cord and the nerves are all in place. All you have to do is excise that cyst and close up the skin, and maybe put a bone graft in there to protect the nervous system. Everything's fine. If you have, yes. Sorry, I pressed the button. Maybe when I hit the PC, it turned it off. Okay, please let me know in the back if you are, nobody back there complained. Okay, sorry about that. Okay, so in between the two, you have the cyst with meninges out there. You're going to tuck the meninges back, seal up the skin, put a bone graft in there, protect the spine, everything's fine. You might have a meningeal myelocele. So myelo, nervous system. If some of the nerves are out there, you've got to have a little bit better surgeon because you've got to tuck all the nervous system back in place and then protect everything. And since your nerves have been pulled out there, maybe there's some damage done. Okay, so now you're getting the idea of one of the answers to the question. When do most of the birth defects show up? If they show up before week 4, they're definitely in the embryonic period. And mommy didn't even know she's pregnant. So if she's taking recreational drugs, if she is smoking and drinking, which are not illegal, but they're stupid if you're pregnant, then if she doesn't know she's pregnant, she's still doing those kinds of things. And it's what they call a teratogen. It makes more likely you can have birth defects. Now, some women smoke and drink and do all kinds of things, and their babies come out fairly normal. She's pretty darn lucky. Okay, embryonic period, first 8 weeks. A lot of women cycle irregularly. If they don't have a period every month, they may not know that there's a missing period for 2 months. So then they start figuring, oh, maybe I'm pregnant, and maybe I should check it out. You've already passed the period when most of the birth defects are occurring. So if you're doing stupid things the first 2 months of your pregnancy, guys don't have to worry, but women have to worry. Okay, you don't see anything, so get out your little ruler. What is the size of 30 millimeters? That big. So you don't see mommy getting big here because the baby's only that big. At the end of development at birth, 28 to 36, so 1.5, that's the size. Yeah, now you can see that space there is shoving everything up into mom's diaphragm and shoving her pelvic organs down and no place else to go, so boom, out in front. Now you know she's pregnant. By the way, if a guy comes to you and he looks like he's 9 months pregnant and tells you that's all muscle, no, he's got pre-diabetic problems. Okay, so here, embryonic period, and then the rest of fetal development. So if you're talking about a fetus, that is after the first 2 months of development. Okay, so teratogen, I mentioned that. Teratogen, it could be a drug. It could be something that's prescribed for the mom. A lot of, if you know about thalidomide, it was a muscle or some kind of a relaxant that doctors prescribe to mothers for, I guess, the anxiety or discomfort or something in pregnancy. I forget exactly what it was prescribed for, but it was a prescription medication, and babies were born without limbs. So it was because of that drug that these really severe problems occurred. So it could be radiation. That's why mommies are not given radiation to the abdominal area. If they have to have radiation, then a lead apron is put over them to protect the baby. Could be an infection, that it could be flu, could be some virus, something else. Could be mechanical stresses. What could cause mechanical stress? Come down here, oligohydramnios. So we know amnio is referring to the amnion. Hydra is what? Water. Oligo, you might not know. Anybody know what oligo means? It means a few. It means that there's too little water in the amnion. What does that do? Baby's not floating free. Baby can't move around like baby should be moving around. He may be, or she may be, sitting on their feet, and it may be in a position that's not so good. So what are some of the birth defects due to oligohydramnios? Club foot. Anybody know or seen a person with club foot? They actually function pretty well, but they're walking usually on the outside of their foot. So, or maybe the inside, depends on which way, on what position they were in. Knee hyperextension. So the whole knee, instead of being able to walk straight, it's actually going backwards. So it's not so good for ambulation when you have knee hyperextension. One side, not so bad. Two sides, pretty bad. So just having too little bit water in the amniotic sac can cause the club foot. You can also sometimes get connective tissue developing in abnormal parts of the amnion. Not supposed to be, it's supposed to be nice and clear sac, but sometimes you get like a scar forming, and it can actually constrict part of the baby. The limbs develop normally, but this scar tissue comes in and constricts them and actually amputates the limb. So here you have what's showing, you have the destructive forces of an amniotic band pressing on the wrist, and then by the time the baby's born, no hand anymore. It was there, but now it's completely gone. So again, all of this, the worst time is going to be, well, this kind of thing, that kind of thing, that happens after the baby gets to be restricted in the amnion. This is a myelomeningocele. You're going to see that the legs look abnormal. You look at the back, that obviously is abnormal. You've got this big cyst on the back. Myelo means nervous system is out there in the cyst. Meningio, that means your meninges are out there in the cyst. So dura mater, pia mater, and the whole thing is out there. The whole cord is out there. So pia, arachnoid, and dura are also out there as well as the cord. So failure of the neural tube to close, that's going to happen early on. So what can cause it? It can be deformations, disruption of vascular system, compression due to the connective tissue developing, some kind of shearing force, tearing force. Could be, here you got your teratogens forming your spina bifida. You can have genetic problems, chromosomal problems, trisomy 23, trisomy 21, those kinds of things. Those happen way, way early, and then you get birth defects. And then a lot of things just don't know why. The unknown. Don't know why that happened. Can't figure out why the birth defect occurred, but it did. So this next slide tells you about the types of birth defects. Gives you a little key down here as to what all the abbreviations means. So you look, 1, 2, 3, 4, 5, 6, 7, 8. Draw the line down. This is the end of your embryonic period. What birth defects form after that? Very few things. So ear problems, eye problems, palate problems, and external genitalia. Development occurs later, so you won't see those problems if they're malformed early on. But all the other things, heart problem, not having a wall between your atria, not having a wall between your ventricles, having blood vessels that are supposed to leave your left ventricle, leaving left and right ventricle. So your aorta is carrying both deoxygenated and oxygenated blood. Those kinds of things will develop early. So cleft lip, cleft palate a little bit later. So feminization and masculinization of genitalia occurs early, but the external, this is gonadal, and then external will show up later. Okay, we talked about mesoderm and ectoderm. So skin is going to be ectoderm. Mesenchymal cells are going to form epithelia, and also organ parenchyma. Parenchyma are fill in the areas between endoderm and external mesoderm with other structures. And then stroma is the connective tissue of the organ. These are coming from your mesenchymal cells. Mesenchymal cells, we get to bone, and they're going to form the skull, your vertebrae, fill in the areas between cartilage that has formed in long bones to form the rest of your endochondral bones. So we're going to be coming back to mesenchyme when we talk about bone and muscle and other tissue. So just another way to look at what is occurring. we have a lot of cell migration that's going on. I told you that your teeth are actually coming from your nervous system, at least your odontoblasts are coming to form part of your teeth. The cells in the center of your adrenal gland, they come from your nervous system. You can actually transplant those odontoblasts. They've taken odontoblasts from fish and transplanted them to the back, and then they have teeth growing out of your back. So you could do that to humans, but you might be in trouble doing that. But you take these cells that migrate, and you can transplant them to other areas, and they're going to form what they would normally form anyway. So here we've got neurocrest cells. Where are they migrating? They're migrating out into the gut, and they're going to form parasympathetic input to the gut. So parasympathetic doesn't run your gut, but it keeps your intestines functioning normally. They have their own nervous system. They have millions of nerves all developing on their own. But we need the autonomic nervous system to kind of keep them under control so they don't get too out of it. So also sympathetic nervous system. We've got the dermatomes, so our sensory nerves that go out in kind of a stripe fashion out to different parts of our body. So that is also due to migration. So looking, this is your neural tube, and then we've got your scleratome that is going to have part of your neural arch that will be your peripheral nervous system. It's going to go around the neural tube and form the vertebral body. That's going to totally obliterate the notochord where the bone forms, and you're going to have that gelatinous center where the disc is. And then you also have ribs that are going to be forming in the thoracic area. All right. So take a look at the embryology. It's just an overview right now. You can also look at Martini's embryology. I find him confusing. That's why I put that together for my purposes. I think it might also help you. To know where these cells are coming from. You're supposed to know all about how atoms form molecules, and molecules interact to form organelles, and then your organelles interact to form the cells get together and form tissues. You have more than one tissue, you can have an organ. More than one organ, you have an organ system. That's where we're starting at. We're kind of backing off and talking about the cell a little bit, but we're going to be talking about organ systems throughout 301 and 302. So we're down here after we finish talking about the cell very quickly, just to remind you what you're supposed to already know about the cell. Then we're going to talk about tissue to remind you what you're supposed to know about tissue. And then we're going to get into the organ systems. The first organ system, anybody know what the first organ system is that we're going to study? Huh? Not an organ system. Tissue. Just kidding. So epithelia is a tissue. First organ system. The largest organ system of your body is your skin. Okay, epithelium. These are cells that sit on a surface. That surface has a connection to the external environment. So your skin, the top layer of your skin is your epidermis. The epidermis is actually stratified squamous epithelia. Happens to be keratinized. meaning you have special filaments, intermediate filaments that will hold those cells very tightly together so that when you scratch yourself, you don't get a whole wad of cells under your fingernail. You go inside your mouth and scratch that hard, you get a wad of cells underneath your fingernail. Why? That is stratified squamous, but it's not keratinized. It's not designed to fight frictional forces. It does provide protection against frictional forces, as you know, when you chew something that's got a lot of rough edges on it, but it's not as protective as that. Other exposed surfaces. So up your nose, in your mouth, up your butt, up your urethra, up your vagina if you've got one. All of those are lined by epithelia. Your lungs have a contact, long route out, but they contact the external environment. Inhale, exhale. So there's a channel that connects the external environment to your alveoli. So your alveolar cells are epithelia. Your blood vessels are not supposed to be in contact. The inside of your blood vessel is not supposed to be in contact with the external environment, nor is the inside of your heart supposed to be in contact with the external environment. So those don't have epithelia, they have endothelium or endothelia. Surface of the body, GI tract, also anus, mouth. Some people shoot food up their nose, but normally it's mouth and anus, that's the connection to the GI tract. Your liver is an outgrowth of your small intestine. Your pancreas is an outgrowth of your small intestine. Those are glands that have invaginated from the surface of the intestine and then developed into these huge organs. We've got parotid gland, submaxillary, or submandibular, those are the same thing, and sublingual glands. Those are invaginations from your mouth, and those are your salivary glands. Anything, your sweat glands, those invaginate from the surface of your skin and form the glands still connected to the surface. What is an endocrine gland? Is what? Doesn't reach the surface. Did it develop from something that was from the surface? Yes. So an endocrine gland developed from the surface, epithelia. It invaginated and went into your body, but then it lost its duct. So your sweat glands, they have a duct to the surface. Your endocrine glands lose the duct. So they dump their secretions into the bloodstream. Anytime you have a lot of cells, you're going to have blood vessels going to those cells. Why is cancer so dangerous? Because you got all these freaky cells that are multiplying out of control, and blood vessels are going to them, and that nutrient delivery to the cancer cells is robbing the rest of your body of nutrients. Even though you don't have a metastatic cancer, you can still die because the tumor robs the rest of your body of nutrients. Gets big enough, you're going to die. Okay? Connective tissue is what it implies. It's connecting something to something else. We have physical connections, and then we have fluid connections. Blood and lymph are fluid connective tissues. The others have structural connections. But blood will connect by flowing. Lymph will connect by flowing from different parts of the body. So they tend to fill in spaces. They actually are more important than that. because your connective tissues will secrete things that will allow for your epithelia to look like they do. So they have chemicals that they release that keep your skin looking like skin, and your lining of your intestine looking like the lining of your intestine. You take those cells off their connective tissue and put them in a Petri dish. You can give them all the nutrients that they need, but they're not going to look like they would in your body. They won't be cuboidal. They won't be columnar. They may still look squamous, but they're going to look more like a fibroblast than a squamous cell. They're going to lose that ability to look like what they're supposed to look like. So connective tissue, yes, it keeps things organized and connected to each other, but the cells and the fibers that are secreted have important things on them that keep things looking as they're supposed to. One type of connective tissue, your adipose, that's a connective tissue, stores energy for you. Some of us have more storage than others of you, and that can be a problem. The storage in your abdominal cavity, that fat is different than the subcutaneous fat. It's not so bad to have subcutaneous fat, but intra-abdominal fat secretes hormones. Fat secretes hormones. Did you know that? How many of you knew that fat secreted hormones? Say, less than 5%. The hormones are different. We got fat here that tells our feeding center, you ate enough, you don't need to eat anymore. Some people don't have the receptor for that hormone up there, and they've made rat or mice model of that, and those rats will just eat and eat and eat and become really obese. If they hook them up to a normal rat, the normal rat will get all its nutrients robbed, and it will shrink, shrivel, and die. Even though it is secreting the hormone that should stop the fat rat from eating, the fat rat already has enough of this hormone. Doesn't have the receptor, so he's producing all this hormone, no receptor. He eats, gets fat, any consequences of obesity, shortens his life. But this poor little rat that is attached to his blood vessels actually gets robbed of all its nutrients and it dies. But if the rat has the receptor that can't produce the hormone, then hooking it up to a normal rat cures the fat rat of its problem, because now it's getting the hormone from the normal rat. The intra-abdominal fat, that secretes a whole nother bunch of hormones. Nasty little hormones. Okay, muscle tissue. We got different kinds of muscle tissue. Obviously, skeletal muscle, cardiac muscle, smooth muscle. Those are our three main kinds of muscle, all designed to move things. Skeletal muscle moves our skeleton. It also provides body heat. When you get cold, you shiver. That generates heat, you get your core body temperature back up, you feel fine. So it has some other functions besides moving your skeleton. Smooth muscle controls your sphincters, close your eye, close your mouth, close your anus. Those, you have skeletal muscles that you can physically control, and then you also have smooth muscle that is inside, that's not under your control. You irritate the smooth muscle inside, and you might poop in your pants, or you might urinate, because you're not aware enough to contract the external skeletal muscle, or you're not potty trained yet. So puppies and babies, you put something in here and it pushes something out there, and everything is out in the diaper or out on the floor, because they haven't learned to control their skeletal muscle sphincter. So we're going to talk about smooth, cardiac, and skeletal muscle. Cardiac and smooth more next semester. neural tissue at the end of this semester. So we'll talk about the brain, the different parts of the brain. We'll talk about the pathways in and out, what a neural cell is composed of, and then we'll also talk about all the supporting cells. Actually, you have far more supporting cells than you have neurons in your nervous system. Okay, epithelial tissue. Epithelial tissue sits on connective tissue. We call it basal lamina or a basement membrane. These are not the same thing. Do not use them interchangeably. This physically separates the blood flow, the delivery of nutrients to the epithelia from the cells that are on the epithelial surface. So everything that reaches the epithelial cells has to go by diffusion. I always hope that one semester everybody understands diffusion. Well, hope that this is the semester it happens. Okay, so epithelial cells, you can form glands from those epithelial cells by having them invaginate, form a duct, and then differentiate into some sort of secretory tissue down below. So you have to invaginate first. You have to maintain that system of invagination as a duct to get a true gland, exocrine gland. Exo meaning going to the outer surface. If you lose the duct, you don't lose the gland, it becomes an endocrine gland. So all of the epithelial cells that we're going to be studying, whether they're simple or stratified, if they're squamous, cuboidal, or columnar, transitional, they have different shapes, but they're all avascular. They're all sitting on a basement membrane. They only can get their nutrients by diffusion. The cells have to be tightly bound to each other, otherwise you're going to be losing them. Anytime you have any friction or any mechanical distortion, they just rip off the surface. So you got to have them bound together. I know you all learn mitosis as though the daughter cells separate and they go their separate ways. No, they don't. In epithelial cells that undergo mitosis, they stay stuck together by cellular junction. So they're going to get stuck together by desmosomes, tight junction, occluding junction, maybe in, well, we'll get to gap junctions later too. Okay, epithelia is there to provide protection. Okay, simple squamous, not very much protection. Simple squamous job is diffusion. Get gases across fast, get fluids across fairly fast. But if you have a cuboidal or a columnar or a stratified, you get more protection. So epithelia is there for protection. The thinner it is, less protection. It's usually designed for another function besides protection. So permeability, that would be a simple squamous cell's job. Provide the permeability. Yes, it's protection, but it's also permeable to gases and to some fluids. Some are there to provide sensation. Our epidermis has modified cells, receptors up in the epidermis that provide us with information about what's touching our body's surface. So some are very sensitive to light touch, vibration. Others, you have to have a deeper pressure and a heavier vibration or pain, some kind of stimulus that causes pain. Or temperature. So we have various nerves that will connect to the brain. Some information travels fast, some information kind of slow. What is faster, pain or temperature? Temperature is faster? body pain. Pain from the surface of your body travels fast. You stub your toe, you're dancing around, you're screaming, somebody sticks a pin in you. You know not only specifically where they stuck the pin in you, but you're also turning around and punching them out. If it's coming from your internal organs, you're not so clear about the pain. It's dull, aching, and kind of diffuse, and you don't really know where it is. And sometimes your brain says, oh, it's coming from your arm. And it's not, it's coming from your heart. So you get some confusion there. But sensation is part of your epithelia job. Okay, I got 5 more minutes. Okay, so we're taking a section through a cell that is taller than it is wide. So that is a columnar cell. So we cut it, and that's what's showing, that's shown there. This is where the plane of section is, and then we're going to look at it. So from above, it looks kind of like simple squamous epithelium, but if you cut down, you see that it's taller than it is wide. Columnar epithelium has a polarity. That means it looks different at the top than it looks at the base of the cell. So what's at the base of the cell? The nucleus. We also have some invaginations at the base. So down here you've got the connective tissue that is really heavy collagen, and then you've got a basal lamina here. We've got actually 3 layers to the basal lamina. So basal lamina, as opposed to a basement membrane, basal lamina has a lamina base, also called lamina basalis, has a layer that light comes through, has 2 layers that light comes through very easily. They're called lamina lucida or lamina rara. Come back here. We've got a lamina lucida, and then they point out a lamina densa, which light doesn't come through very well. But there's actually another layer in there that your textbook isn't mentioning, and that's the lamina rara interna. So we've got an externa that's next to the epithelial cell, and one that is next to the connective tissue. The basal lamina is secreted by the epithelial cells and the fibroblasts that are forming the connective tissue down below. So it's co-secreted. This is why it's so important for maintaining the shape of the epithelium, because the epithelial cells control what chemicals are attached to these connective tissue fibers in the basal lamina. If they lose those chemicals and just have agar, they're not going to be able to attach the chemicals they need for the differentiation. So they lose their shape when they're removed from the basal lamina. With lamina rara interna, deepest in, lamina rara externa, out next to the epithelial cells, and then a lamina densa sandwich in between. So actually those 3 layers. If you add a reticular lamina to those 3 layers, then you have a basement membrane. So you need this 4th layer to get a basement membrane. Most of the time you don't have this 4th layer. If it's there, then you have a basement membrane, or BM. If you don't, you have a BL, basal lamina. So again, if you take the cells and put them in a petri dish on agar, they don't have the chemicals that keep them looking like they're supposed to. So those columnar cells, they flatten out and look like fibroblasts. So deepest in, you have stem cells. All epithelia turn over at a high rate. So they are going to be sloughed off and have to be replaced. So stem cells people who are opposed to using fetal stem cell and or don't have access to fetal stem cells, they're looking for other stem cells in other areas and trying to get those stem cells to do what fetal stem cells do, with some success and some not so successful. Okay, so you can see I'm already behind. So Wednesday we'll pick up and finish